PhyNexus is a life science tool company providing innovative and integrated high-performance purification tools for proteins and plasmids. In this talk, we present a type of high-performance immunoprecipitation that is performed on a pipette tip column. We call the pipette tip column a PhyTip column. The PhyTip pipette tip column has been designed to outperform all other immunoprecipitation techniques based on bulk beads and magnetic beads. The PhyTip column is a basic technology that is used on Rainin pipettes and automated robotic liquid handlers. Essentially, we perform chromatographic processes on a pipette tip. Affinity resins are placed at the end of a pipette tip. Low back pressure and low dead volume screens facilitate high performance capture, efficient washing, and high concentration elution of materials of interest. The technology is flexible with choice of tip size, choice of resin bed volume, and choice of resin type. The cornerstone of the technology is our back and forth flow of sample and buffers through the tip of the column. This back and forth flow liquid motion is unique to chromatography and is responsible for producing a tip concentrating effect. This allows the recovery of very high concentrations of pure protein even from limited sample. The sample material being captured containing a target protein or protein complex of interest passes back and forth through the column until the capture of the target protein is at equilibrium with the resin. That is, the capture process is complete. The back and forth flow ensures that the capture is concentrated on the end of the pipette tip, even if the column bed is very small, and even if the sample is very limited. Because the sample is captured in a very small bed volume, and the elution is into a very small liquid volume, the tip concentrating effect transforms the immunoprecipitation process into a high-performance process. The tip concentrating effect of the technology produces a much higher concentration of product, and the end product is much purer. Further, because of the tip concentrating effect, we can miniaturize the process. This results in minimal use of expensive reagents. For example, we use much less of the antibody reagent to capture the target protein of interest. This saves significant money. We use computer-controlled semi-automated and automated instrumentation, which enables higher throughput parallel operation, rapid protocols, and reproducible protocols. There are two ways that HPIP can be performed: a direct method and an indirect method. Direct HPIP is where a specific antibody is first loaded onto the resin in the PhyTip column to make an activated column. Then the target protein from the sample is captured by antibody bound to resin in the tip. A wash is used to remove nonspecific bound material and purify the complex. Finally, the purified enriched antibody protein complex is eluted from the PhyTip column. Indirect HPIP is where a specific antibody is mixed with target protein in the sample in bulk solution. The antibody protein complex is formed over a period of many minutes or a few hours. Once the complex is formed, the sample is processed by the PhyTip column to capture the complex containing the target protein. The column is washed to purify the complex. Finally, the purified enriched antibody protein complex is eluted from the PhyTip column. We will demonstrate both of these processes in this presentation. This is a graphic depiction of the beads packed into a pipette tip column. The figure shows an enlargement of a column bed filled with affinity beads and the fluid surrounding the beads. In order to examine the HPIP process, Let's examine the processes that occur in the fluid and resin bead and at the fluid-bead interface. The direct HPIP process is as follows. An antibody is raised against an antigen protein and produced to be used as a reagent. This protein is applied to protein A or protein G PhyTip column. The column resin captures the antibody and the excess antibody is washed away. The column is now ready to capture the target protein. A sample containing the target protein is applied to the column. The target protein is depicted as the blue dots. Other contaminant proteins are other colors. The resin captures the antigen with the antibody that is bound to the resin. After washing nonspecific proteins away from the resin, the antibody and target antigen protein are eluted from the resin and analyzed. This slide shows the indirect HPIP process. A sample that contains the target protein is provided usually as a clarified cellular lysate. The target protein is depicted as the blue dots. Other proteins are other colors. An antigen that is specific against an antigen protein target, the blue dots in the above picture, is introduced into the cellular lysate. The antibody binds to the target antigen protein. Then the sample solution is applied to a protein A or protein G PhyTip column. Instead of capturing the antibody alone, the resin captures the antibody, which in turn is bound to the antigen target. After washing nonspecific proteins away from the resin, the antibody and target protein are eluted from the resin and analyzed. This slide shows how we designed experiments to test and compare HPIP. The antibody-antigen complex was eluted with 15 microliters of buffer. The recovered samples were separated by SDS page analysis and detected with a silver-stained gel. The experiment design included controls and standards. If the antibody is successfully captured by the column, we would see two bands, one from the heavy chain of the antibody and the other from the light chain of the antibody. If the antigen or the protein of interest is also collected, we would see three bands, two from the antibody, heavy and light chains, and one from the target protein. Direct HPIP was carried out and compared with Dynabead magnetic beads. Pluses and minuses indicate the inclusion or exclusion of antibody, antigen, protein, or both within a given sample. The time needed for the protocol is noted beneath the gel. With HPIP, the antigen target protein is recovered in much higher concentrations compared to magnetic bead recovery of the antigen target protein. The HPIP takes a little less time, 55 minutes compared to 69 minutes for the magnetic bead process. Now, indirect HPIP was carried out and compared with Pierce bulk beads. Again with HPIP, the antigen target protein is recovered in much higher concentrations compared to bulk bead recovery of the antigen target protein. In this comparison, the HPIP technology takes significantly less time, 30 minutes compared to 190 minutes for the bulk bead process. This slide compares direct HPIP with indirect HPIP. Direct HPIP is generally preferred because it has a simpler experimental design and the overall method is fast. No overnight incubation of the sample is needed, and with this particular experiment, higher concentrations of the target protein were recovered with the direct method versus the indirect method. The indirect method is preferred when the binding kinetics of antigen protein and antibody are slow. or when there is concern that shear forces could disrupt a large or fragile protein complex. In summary, HPIP is characterized by experiments that are automated and processed in parallel. It is cost-effective. It is high performance. Also, it is worth noting that the technology can be applied to other types of chemistries. For example, co-IP can be used for large protein complexes. Tagged proteins such as His-tagged protein can be used as protein baits for capturing protein complexes, and chip experiments can be used to study DNA-protein interactions. See your PhyNexus representative for details on these methods. And thank you for your attention.